Across species, big animals tend to age more slowly than small animals. Within species, smaller individuals tend to live longer and appear to age more slowly. But I think one question that's worth asking is this really slower biological aging? One of the holy grails of longevity research today is what are the mechanisms that underlie species differences in longevity and aging rate? Pretty intriguing data suggesting that into people, there is this mechanistic connection between the levels of growth hormone and IGF-1 signaling during development, adult body size, and rate of aging. My name is Matt Caberline, and welcome to the Optispan YouTube channel. Hey everyone, welcome to the Optispan podcast. And on today's episode, we're going to cover a topic that I see a lot of people in the field, including a lot of experts, get wrong. And that topic is the relationship between body size and aging. And this is really, I think, fundamental to the biology of aging. So after watching this video, you will be able to show off your longevity knowledge. And if you're ever in a situation where you hear one of the experts get this wrong, you'll be well positioned to correct them if you so choose. So the relationship here is actually pretty simple. And the key differentiator is whether or not we're talking about different species, looking at the relationship between body size and longevity across species, or we're talking about individuals within the same species. And that's the key thing to pay attention to. So if we look across species, and I think this is true in general across the animal kingdom, certainly within vertebrates, it is the case that larger species of animals tend to live longer. They have longer median and maximum lifespans, and they appear to age biologically more slowly than smaller species. And we'll come back and we'll look at a few of the differences here. But if you just think about like dogs to humans, the difference between the species, right? Dogs are a different species than humans. Dogs age biologically about 7 times faster than humans, and dogs on average are 7 to 10 times smaller than humans are, okay? So again, bigger species tend to age more slowly and live longer. Now, if we look within the same species, so if we only look at humans or only look at dogs, at an individual level, bigger individuals tend to age more rapidly and live shorter lifespans. And again, dogs are nice here because the difference is pretty obvious, right? So anybody who's familiar with a Chihuahua and a Great Dane will recognize, first of all, that a Chihuahua is much smaller than a Great Dane in terms of body size. And their life expectancy is actually about twice that of a Great Dane. And if you look across all of the different breeds of dogs, this correlation holds up quite nicely, that big individuals within the same species tend to age more rapidly and have a shorter lifespan. Okay, so I'll come back to that, but that's the key take home, right? Across species, big animals tend to age more slowly than small animals, or at least have a longer lifespan than small animals do. And within the same species, larger individuals tend to have a shorter lifespan and appear to age biologically more rapidly. Okay, so where you'll hear people get this wrong is they don't understand this differentiation between within species and across species. So you hear a lot of people say things like, big animals age more slowly than small animals, except for dogs. Dogs are this weird outlier, and that's not the case. In fact, if we look within species, we see this relationship in mice and in people as well, that smaller individuals tend to live longer and appear to age more slowly. So that's where the mistake comes in. And if you hear people make that mistake, now you can correct them. Okay, so why do so many people get that wrong? And I think it's because this relationship within species is just much more apparent in dogs, right? Again, if you look across the breeds, the difference in body size between a Chihuahua and a Great Dane is something like tenfold, right? We don't have tenfold differences in body size within people or typically within mice. And so that difference in body size isn't as apparent, and the magnitude of effect on longevity and rate of biological aging is just smaller, and so it's harder to see. But it does exist, and again, we'll get into some of the data indicating that in a little bit. Okay, let's focus first on this relationship across species. Again, in the animal kingdom, across species, big animals generally live longer than small animals, okay? And you can see this pretty easily if you look within vertebrates. And one of the things that's interesting here is that this relationship is linear on a log-log plot. And what I mean by that is if you plot the logarithm of maximum lifespan against the logarithm of body mass, it becomes a linear relationship, and there's a strong linear correlation. And so, again, you can look from very small animals like rodents, shrews, mice, rats, all the way up to very, very large animals like whales. And across that whole spectrum, you have this nice linear relationship. Now, of course, there are species that are above or below the linear regression line. And it turns out humans are above the linear regression line, along with species like naked mole rats or bats. And I think one of the interesting questions here is there something that's kind of unique to these species that tend to fall above the line that's a little bit different about the way they're aging compared to other species that are right on the line or below the line? And I think we don't know the answer to that. But again, even with these outliers in there, you can see this nice, strong linear relationship on a log-log plot between maximum lifespan and body size. And it's pretty big. Again, it's worth appreciating. The magnitude of difference here is multiple orders of magnitude, going from very small animals all the way up to the very, very large animals. So again, think about a mouse that lives, a couple of years on average compared to a whale that might live a couple of 100 years. That's 2 orders of magnitude. And we could get even down to smaller animals like fruit flies and nematode worms that live on the order of a few weeks. So again, we're looking at 100 to 1,000-fold differences in species lifespan when we look across species. And I'm making a big deal of this because when we look within species, that magnitude of difference is actually much, much smaller than it is when we look across species. Okay, so there's a strong correlation between maximum species lifespan and adult body size on a log-log plot. And again, this has usually been done by looking at maximum species lifespan because for a lot of these species in the wild, we don't really have good data on median lifespan of those species. But I think one question that's worth asking is this really slower biological aging? And that's hard to answer with 100% confidence, but it really does seem that way. Where this has been studied at the organ level, tissue level, and even at molecular level, looking for the hallmarks of aging, it really does seem to be the case that in these larger species that are living longer, they are also aging biologically more slowly. And that can also be seen with some of these newer technologies that have come online, like the DNA methylation tests. We're starting to see those same signatures where it really does seem as though many aspects of biological aging are happening at a delayed rate or a slower rate in these very, very large animals compared to the small animals. So I think with a pretty high level of confidence, we can say that many aspects of biological aging are different and happening at a different rate. So it really is as if the aging process has been slowed or sped up as you go from large animals to small animal species. Okay. So one important question, of course, is what's the mechanism that accounts for this? And I would say, unfortunately, we don't know. And in many ways, this is one of the holy grails of longevity research today is what are the mechanisms that underlie species differences in longevity and aging rate. And in part, that's really important because the magnitude of effects are so great. Again, as I was already talking about, 100 to 1,000-fold differences in longevity looking across species. If we could understand how that's working, obviously that would open up all sorts of potential areas for therapeutic development. So a lot of people are interested, but it's really hard to study because of how long these very long-lived species live. So it's difficult to do the sorts of mechanistic studies you would really want to do to try to figure out exactly what's underlying these dramatic differences in aging rate. And I would say, for now, the mechanistic explanations that have been put forward are pretty unsatisfying. So, one idea is this idea of rate of living, that metabolic rate is underlying the differences in species lifespan. Smaller animals tend to have higher metabolic rates, especially when you normalize to body mass. Another idea that's been put forward is that genome maintenance and DNA repair differences account for some of these species level differences. And there's some evidence to support that. Some of the very long-lived species have different ways of enhancing their ability to maintain their genetic information and to reduce mutations. Whether that's cause or not of their longevity is challenging to prove. And in fact, you could say that it is maybe necessary but not sufficient to have higher genetic fidelity. In other words, very large animals have a lot more cells, a much higher risk of cancer, just simply because of the number of cells. And so they have to evolve these enhanced DNA repair mechanisms or anti-cancer mechanisms in order to make it to adulthood, to reproduction. But is that causing their extreme longevity? Again, that's a little bit harder to tease out. So I don't know. I'm kind of skeptical that genome maintenance is sufficient to account for the extreme longevity of some of these species. People have also put forward evolutionary explanations. So, one idea is that smaller animals tend to be subject to predation. So they have to reproduce very rapidly, and there's no reason for them to evolve later reproduction and these longevity assurance mechanisms to make it into very, very long lifespans. I think that's reasonable. It doesn't tell us much about the mechanisms of longevity. Another related idea is that larger animals tend to reproduce later and they have fewer offspring. And so they need to invest more resources into longevity in order to care for those offspring, get those offspring to adults so that they can then, pass their genes on to the next generation successfully. Again, I think that makes sense. It just doesn't really tell us anything about what are the actual mechanisms that are at play here. It's more of an explanation for why they need to develop these mechanisms of slower aging and enhanced longevity. So again, I feel like it's kind of unsatisfying. I think one interesting piece of information that has come to light recently, again, looking at comparative studies of dogs to humans, is while larger animals tend to age more slowly than smaller animals, it's not necessarily a linear relationship between the differences in rate of aging across the entire life course. And so again, if you've talked to veterinarians or if you've been to a veterinarian's office that has these charts where they show dog age versus human age, and they usually have small, medium, and large sized dogs. The idea, of course, is that bigger dogs are aging faster than small dogs, but it's not this 7-to-1 relationship that as kids we're sort of taught. 1 human year is about 7 dog years. So that varies based on the size of the dog. But when we look at a molecular marker of aging rate, in this case, DNA methylation, so these are the epigenetic aging clocks that a lot of people are enthusiastic about these days. If we look at that and we compare the epigenetic rate of aging of a dog versus a human, it's not 7-to-1 over the dog's lifespan. It's more like 15-to-1 early in life, and then as the dog and the human are getting older, it kind of tails off, and maybe it's closer to 2-to-1 or 3-to-1 out towards the end of the dog's life. And there was a really neat paper from Elaine Ostrander and Trey Eidecker a few years ago that showed this, and I really like this graphic where they have Tom Hanks on the y-axis representing human years and a dog on the x-axis representing dog years, and you can see this nice non-linear relationship between the rate of aging in dogs and the rate of aging in humans. And so I think it's unclear whether this non-linearity is going to be the same if we were to compare, mouse aging to whale aging. But it's intriguing that at least in this case where we do have a direct comparison, you do see this interesting non-linearity. And it suggests that what's happening early in life is maybe driving a lot of these species differences in aging rate as opposed to what's happening later in life. And that's interesting because, as you'll see in a minute, it also seems to be things that are happening during development that drive much of the intraspecies differences, the individual differences within the same species in terms of aging rate. So development seems to be particularly important maybe in both of these cases. Okay, so that's looking across species. Now, what about within the same species? And again, this is where a lot of people get confused and they think dogs are sort of this weird case, but in reality, I think this relationship is preserved within species, at least for many animals, if not for all animals. And that relationship, again, is now flipped from what we were just talking about across species. Within species, smaller individuals tend to live longer and appear to age more slowly. And it's important here to recognize what we're talking about is adult body size controlling for things like obesity. So obesity obviously is something that can affect longevity, and I would say there's reason to believe that obesity may accelerate many aspects of biological aging, but that's not what we're talking about. We're talking about adult body size independent of abnormal weight or obesity, okay? So this would be typically measured by height in a human or length in certain animals, but not necessarily body weight or adiposity or body composition in any way, okay? So really we're talking about the frame of the adult individual. And adult body size is largely determined during development, and it's determined by a combination of genetic, epigenetic, and environmental factors. And I think it's safe to say that perhaps the most important environmental factor that is going to determine adult body size is going to be nutrient status, how much food is around and what is the type of food that's around. And perhaps the most important environmental factor is nutrient availability, and in particular protein availability. And again, the amount of food that is in the environment during development seems to play a particularly significant role in determining adult body size at the individual level within a species. So one maybe useful way to think about this is genetics will set kind of the upper bound on what an individual's adult body size can be, and environment and food in particular is really what determines how close to that upper bound an individual will come. And I think related to that, another way to think about this is that adult body size is determined by some key hormones and their abundance during development. And these, again, are going to be hormones that are influenced a lot by nutrition. Things like growth hormone and insulin-like growth factor 1 play a major role in determining body size during development. And so again, levels of these hormones are influenced by genetics, and that can sort of set the maximum achievable upper bound, but then they're going to be profoundly influenced by the environment during development. That could be in utero during pregnancy, or it could be during early formative time of development post-pregnancy. Both of those things can play a role in the determination of adult body size. Okay. So again, I think let's start with dogs, because that's something that most people are familiar with, and most people are familiar with the idea that big dogs age faster than small dogs. Again, go back to the Great Dane Chihuahua example. A Great Dane is going to age about twice as quickly as a Chihuahua will, has about a 10-fold difference in body size. So if we look across all breeds, we see this is a nice linear relationship again, but here it's interesting to note it's not a log-log plot, it's a linear plot. So if we plot average breed lifespan on the y-axis and adult body size or body mass on the x-axis, we get a pretty reasonable linear correlation. And again, I make this point because when we look across species, remember, it's a log-log plot. When we look within species, it's a linear plot. And again, I don't know what that means at a biological sort of explanatory level, other than the mechanisms appear to be different, right? And that's not shocking, but it's worth appreciating that this relationship also is mathematically different. And as is the case when we look across species, it seems to be not only lifespan that is influenced by body size, but rate of aging. Again, think about dogs. Big dogs seem to age biologically faster when we look at many different tissues and organs, functional declines, diseases of aging, compared to small dogs. That's true for arthritis and kidney disease and heart disease and cancer in particular. We see all of those things happen at an earlier chronological age in large dogs compared to smaller dogs. I think the one interesting example that runs counter to that, at least in dogs, is dementia. So from the Dog Aging Project data, it seems like big dogs do not show age-related cognitive decline at an accelerated rate compared to small dogs. And so that's interesting, and it suggests to me that there may be something different about aging in the brain versus aging in the rest of the body. And again, my speculation is it comes back to these same hormones that I was alluding to earlier. Growth hormone, insulin, IGF-1 signaling in the brain may have a different impact on cognitive function and dementia in the brain compared to in the rest of the body. But that's just speculation at this point. Okay, so as I alluded to earlier, many people seem to think this relationship is unique to dogs, and it's not. So this has been seen in mice. Multiple studies have seen in wild-type mice, if you look at the individual level, that bigger individuals have a shorter lifespan in mice. One paper from Rich Miller, who is one of the leads of the NIA interventions testing program, was titled "Big Mice Die Young, Early Life Body Weight Predicts Longevity in Genetically Heterogeneous Mice." For you, those of you who are mouse longevity aficionados, this is using the Het3 strain that the interventions testing program uses, but other studies have seen this same relationship. So in mice, it seems to be the case that smaller individuals live longer. And this is also true in humans. It's a little bit tricky in humans, in part because there are social consequences and potentially economic consequences that go along with height. So again, in general, I think it's recognized that taller height is associated with some socioeconomic advantages compared to shorter height. And so that complicates things in people because, of course, we know that socioeconomic status will have an impact on longevity. But even considering that, multiple studies have seen that smaller individuals tend to live longer and tend to have lower rates of multiple age-related diseases when comparing by chronological age. So it really does seem to be the case that small individuals in humans also age more slowly than large individuals. It's just that that delta is pretty small and probably countered somewhat by the socioeconomic complications associated with height in humans. Okay, so what's the mechanism here for the within species variation in longevity? Again, remember, which is opposite that we see between species. And I think here we have a pretty good idea of what's actually underlying this. The most plausible explanation is that the same hormones that determine body size during development are driving a faster aging rate. And in this case, specifically, we're talking about growth hormone, insulin-like growth factor 1, and our old friend mTOR, all of which act in a pathway or something called the somatotropic axis to determine body size during development. They also have other roles, obviously, post-developmentally, sexual maturation, muscle function, muscle growth, repair, during adulthood, but they're really important during development at setting body size, adult body size. So high levels of these hormones during development will cause an individual to grow bigger and have a larger adult body size. Low levels result in smaller adult body size. And so the way this works is that growth hormone is produced in the pituitary gland. That signals, among other things, to the liver to produce insulin-like growth factor 1. And then insulin-like growth factor 1 promotes downstream growth targets, including mTOR, to help set body size during adulthood. So again, during development, nutritional status is really a key driver of growth hormone and IGF-1. And so that's the connection here between nutrient status during development, these pituitary hormones in the form of growth hormone signaling to IGF-1, promoting growth throughout the tissues and organs of the body. And one thing that's really interesting to note here that supports this idea is when we look at dogs, Elaine Ostrander showed back in 2007 that the single largest genetic determinant of body size in dogs is the IGF-1 gene. So a single allele of IGF-1, so a single variant in that gene, is what determines much of the body size difference across breeds in dogs. And the correlation goes the right way. So smaller dogs have lower IGF-1 levels during development. Larger dogs have higher IGF-1 levels during development. And of course, as we've already talked about, larger dogs age more rapidly than small dogs do. So again, all the arrows are at least going in the right way. So that's pretty strong correlative evidence to support this idea that growth hormone IGF-1 levels during development are really what's driving differences in aging rate within species. But what about causality? And I think the first clear example of this was work that Cynthia Kenyon did back in the early 1990s, where she showed that you could actually, in a targeted way, make a mutation in the worm version of the IGF-1 receptor. It's called DAF2. It's the insulin IGF-1 receptor in worms. You make a mutation in that turns down signaling, and that doubles the lifespan of the worm. So this was done in Caenorhabditis elegans, the nematode worm that we've talked about before on the podcast. So a single mutation in this case is sufficient to cause a doubling of lifespan by reducing signaling through the insulin IGF-1 pathway. And so that clearly demonstrates causality of this pathway to modulate aging, at least in a very simple animal. And then since then, many studies in rodents have shown that at several points in this growth hormone IGF-1 pathway, you can make mutations that will increase lifespan in mice. So a couple of famous examples are the Snell dwarf and the Ames dwarf that were very well characterized for their effects on longevity by Andy Bartkey and Holly Brown-Borg and others. These animals are very interesting. They're both caused by mutations that affect pituitary function and growth hormone secretion by the pituitary, so pretty high up in the pathway. They result in animals that, as you might guess by their names, are dwarfs. So they have very small body size, very low growth hormone, very low IGF-1, and very, very large increases in lifespan. So 50, 60% increases in lifespan. Multiple studies have seen this. So again, supporting the idea that not only in this simple nematode worm, but in a more complex mammal like a mouse, turning down these hormones during development is sufficient to drive very large increases in lifespan. And since then, many, many components of this pathway have been found to have similar effects. So there was a nice 2018 review article titled Mouse Models to Disentangle the Hallmarks of Human Aging that actually points out multiple components of this pathway that have been shown to have a positive impact on longevity when we reduce signaling through the pathway. So it seems pretty rock solid that in mice you can turn down growth hormone IGF-1 signaling during development, and that's enough to result in enhanced longevity. Now, it's probably worth saying these increases in lifespan come with some costs. So the dwarf mice that I mentioned, for example, wouldn't survive on their own. They certainly wouldn't survive in the wild. And in fact, even in the laboratory, they are typically housed with wild type litter mates for warmth, right? Because these animals can't actually, the dwarves can't actually thermoregulate well enough to maintain their body temperature. So these are mutations that can lead to dramatically increased longevity, but in the real world, probably wouldn't, because the animals wouldn't survive through development to reproductive age. So again, like many longevity interventions in the laboratory, these come with pretty significant fitness costs. So what about humans? And again, we talked about the correlations that exist in people between body size and longevity. I think one really interesting example here are the little people of Ecuador. These are a population of individuals that have a mutation in the growth hormone receptor. It's a condition called Laron syndrome. And several years ago now, Walter Longo and colleagues who've been studying these people for many years published a paper in Science Translational Medicine titled Growth Hormone Receptor Deficiency is Associated with a Major Reduction in Pro-Aging Signaling, Cancer, and Diabetes in Humans. And what they did was they studied these people in Ecuador and compared them to roughly genetically matched population of individuals who don't have the growth hormone receptor. So these little people, as you might guess by their name, have dwarfism. So they are very short. They have very small adult body height. And that's because of the reductions in growth hormone and IGF-1 through development. They have very low levels of growth hormone and IGF-1 into adulthood. And interestingly, they have very low levels of multiple age-related diseases. So in this initial study was that the little people within this population had much lower risks of cancer and metabolic disease compared to the normal height controls. And since then, they've gone on and also showed, I believe, much lower rates of dementia in this population. I think the interesting thing here was that despite these reductions in at least a subset of age-related diseases, the longevity of the little people was no different than the longevity of the normal height controls. And that seemed to be accounted for by a pretty significant increase in deaths due to alcohol-related diseases and accidents. And so again, I don't know exactly what that means, but I suspect this is pointing to some of the social challenges that come with very, very small body size in human populations, and that might lead to offsetting consequences that mask what could be a pretty significant increase in longevity due to a reduction in at least a subset of the major killers among the age-related diseases. Nonetheless, I think this does suggest that in people, significant reductions in growth hormone signaling and IGF-1 during development are associated with a pretty dramatic reduction in multiple age-related diseases, consistent with the idea that these individuals are in fact aging biologically more slowly than people with higher levels of growth hormone and IGF-1. So pretty intriguing data suggesting that, all the way from very simple nematode worms to mice and into people, there is this mechanistic connection between the levels of growth hormone and IGF-1 signaling during development, adult body size, and rate of aging within species. Okay. So then I think, one last question that's kind of interesting is I talked about, and I kept emphasizing the importance of these hormones during development for defining adult body size. What would happen if we could turn down the signaling of these hormones specifically during adulthood? Would that be enough to give you the longevity benefits and the benefits in terms of reductions in age-related diseases? And it's a complicated question, and I don't think we actually know the answer to that yet, but it's important for a bunch of reasons. In particular, one, I think, thing that has been interesting to those of us who study the biology of aging, preclinically in laboratory animals, is this phenomenon where in human medicine, particularly in the so-called anti-aging medicine category, one of the staples therapeutically for decades has been to give older people growth hormone to boost their growth hormone levels. Again, the idea being that many elderly people have a decrease in growth hormone. In fact, most people as we age will show a decrease in growth hormone. And that could be contributing to things like sarcopenia, loss of muscle mass, reduced bone density. And so restoring growth hormone levels to youthful levels might be beneficial. Intuitively, it makes sense. And so this has been a therapeutic strategy that has been employed in the anti-aging medicine world for quite a long time. It was always a little bit perplexing, though, because that's the opposite direction to what we see increases lifespan in mice. And as I mentioned, has some benefits in terms of age-related diseases, at least in humans with growth hormone deficiency. Again, it's a very different situation, though, having reduced growth hormone during development versus having low growth hormone, low IGF-1 post-developmentally. Okay, so that was always a little bit perplexing. And again, I would say we don't know the answer as to whether growth hormone therapy for older adults is beneficial. My take on the literature is that it probably really has very little in the way of beneficial effects for most people. It doesn't really seem to move the needle much in terms of at least muscle function into old age. So the flip side of that, though, is there's a biotech company called Loyal, which is developing therapeutic to reduce IGF-1 levels, specifically in older dogs, with the idea that turning down IGF-1 signaling might actually increase longevity in dogs. And again, their strategy is to do this post-developmentally. So we have sort of two opposite approaches, both with the idea that they might be beneficial in terms of longevity and age-related diseases. So what data do we have to draw on to kind of help us figure out what's the current lay of the land here. And unfortunately, there's not a lot. There's one study that I think is pretty interesting in mice that comes from Derek Huffman and colleagues at Einstein Medical School. And what they did was they used an antibody intervention to reduce circulating IGF-1 levels, specifically in middle-aged mice. And I would say the results are intriguing, but a little bit mixed. So what they found was that if they reduced IGF-1 levels in middle-aged mice, that was sufficient to increase lifespan by about 9%, but it was only in female mice, so there was no effect on the male mice. So it was statistically significant, but it's not a huge effect, and it's really nowhere near the magnitude of effect that was seen from other interventions that reduced growth hormone and IGF-1 signaling during development. There we're talking 30, 40, 50, even 60% increases in lifespan. So a much smaller magnitude of effect and a sex-specific effect on lifespan from turning down IGF-1, at least using this method during middle age in mice. So again, let's take a look at the data. Those of you who have been watching the podcast will start to get familiar with my sort of rule of thumb for figuring out, does this look like a real effect? 9% is pretty small for mice. So you really want to ask the question, do we believe that this is real? So the approach here is to first look at the lifespan of the control strain. We want that to be around 850, 900 days optimally. So the way we do that is we look at the survival curve. We go to 50% or 0.5, depending on how it's plotted on the y-axis. We go straight across to the control curve, and then we go straight down, and we see where does that hit the x-axis. In this case, it hits the x-axis at about 840 days. So that's not terrible. That's kind of right in the ballpark of where we want it to be. Maybe it's a little short, but it's not like many of the studies where you'll see the controls come in at 500, 600 days, and you're like, I can't believe anything from these studies. So this is pretty reasonable. Then we can ask, where's the median lifespan of the treatment strain, the long-lived strain here? Same strategy, start at 50%, go over to the treatment curve, go straight down. In this case, 952 days. So that's pretty good. Again, that's a reasonable lifespan for a 10% effect. So it seems pretty reasonable. I obviously would love to see this reproduced, but I don't have any reason to be super skeptical of this result. And given what we know about this pathway and its effects on cancer, it kind of makes sense that turning down IGF-1 in adulthood, even if it's not fundamentally affecting the rate of biological aging, could have an effect on longevity, particularly in mice, which are very cancer prone, just from the expected effects of reducing cancer. So I think it's real, but again, it's nowhere near as impressive as what we would expect to get if we did this intervention throughout life, starting in adulthood, resulting in a smaller adult body size. Now, the thing here that was a little bit concerning to me was that when you look at body weight, the females who had their lifespan extended did show a slight reduction in body weight, but that reduction in body weight was all lean mass. So there was actually a shift in body composition towards higher adiposity, higher percent body fat. Clearly, it didn't impact the lifespan, or it didn't prevent us from seeing a relatively small lifespan effect in these animals. But this is something I would be concerned about if we saw that same shift in body composition in middle-aged and elderly people, where we know that in humans, probably more so than in mice, maintaining muscle mass, maintaining bone density is critically important for health span going into older age. I'd be very concerned about an intervention that reduced lean mass, potentially reduced bone density. I don't think they looked at bone density in this study, but you would maybe expect to see a reduction in bone density as well from lower IGF-1 signaling. So this is a little bit of a red flag from a therapeutic perspective about thinking about trying to translate this directly into humans. So at this point, I'd say it's likely you can get a small increase in lifespan from reducing IGF-1 and growth hormone signaling post-developmentally during middle age, but it's probably nowhere near as robust as the effect of doing that throughout development, which in that case is also going to result in a significantly smaller adult body size. However, those effects in middle age may be sex-specific. Again, I don't think it's been studied enough in mice to know whether that was a strain-specific thing or whether this is a IGF-1 thing or whether this is a general phenomenon where it's only seen in females and not in males. But I'd be cautious about interpreting this and extrapolating to humans where we might be more worried about the body composition changes. So maybe okay for a mouse, probably not great for a person if those body composition changes were also translated to people. And so related to this are some concerns around the idea of a low protein diet in middle-aged people or in elderly people. Again, protein is going to be one driver of IGF-1 levels, and there's pretty good data that people on a very low protein diet can see reductions in IGF-1 levels. Calorie restriction across the board will also reduce IGF-1 levels, probably reduce cancer risk, but again, in middle-aged and older people, potentially also reducing lean mass, reducing bone density, and shifting body composition towards more adiposity on a percent basis. Again, some concerns about the effects there on frailty and sarcopenia and fracture risk that, at least personally, I'm not comfortable with that as an outcome where we would see lower lean mass, lower bone density, likely increased risk of fractures and frailty. So just something to be aware of as we think about how protein levels, nutrition are interacting with growth hormone and IGF-1 levels and some therapeutic strategies around that. Okay, so to summarize, across species, larger animals tend to live longer and appear to age biologically more slowly than smaller species. But within species, that relationship is flipped, and smaller individuals tend to live longer and appear to age biologically more slowly than larger individuals. Across species, we really don't have much clue as to what the mechanisms are there, but within species, there's pretty good reason to believe that this is at least largely mediated by signaling through developmental hormones, particularly growth hormone, IGF-1, downstream signaling to mTOR, which are influencing adult body size and rate of aging throughout life, which is how you get some of these larger effects on longevity in the mutants that have very, very significant reductions in these hormones during development. Okay, so I hope that was clear. I hope this has been informative. And if nothing else, at least the next time you hear an expert get up there and make the mistake about dogs being unique, you can correct them if you so choose. As always, if you have any feedback, questions, comments, please leave them below. If you're not already a subscriber, please consider subscribing. And I look forward to seeing you next time on the Optispan podcast.